Okay, good morning, everybody. Any questions or concerns before I get going? Zooming in, feel free to unmute and type up so I know what's going on. We typically don't see if the chat stuff is happening until after I'm already done with lecture. Okay, so a couple quick things. I should have details about the exam, but I really don't have all of that stuff at this point. The proctoring stuff, even though it is all online and apparently AI stuff, it takes time when there are 50 some odd people taking hours to take the test and it has to review all of that stuff. I looked at it this morning and I know people took it, but it shows me 30. I could see the grade book and know that people took it, but it's only processed a little over 2/3 of you so far. So that's all fine and good, but I like to see what's up with the proctoring before I can start spouting off all sorts of stuff. I believe it looks all fine and good. I can see the people that it has proctored. Handfuls of flags, which I expected to see. I only opened up 2 people's thing, but it's like, oh, headphones got put in, or opened a book, opened a tab, which is the book, one or the other, notebooks. It's all stuff that I expect to see, but I still need to review that to see if there's any sort of shenanigans. Hopefully not. So with that said, there are still some issues, and one person that takes the exam elsewhere, I had an issue and didn't put the proper amount of time on there. So that test didn't start, it starts today. So technically I don't have everybody done, but I can tell you at this point for those people that are curious, it looks like the average was a 70 point something. That can adjust the littlest bit, all right? So that's not subject to potentially change slightly, but I expect it to be right around there. That seems pretty good. The scores definitely ranged, and like I said, I don't really feel comfortable saying stuff until I've truly evaluated it. But it looks like some pretty high scores, and it also looks like some pretty stinking low scores, which makes me wonder, you do know there's a textbook. Hopefully, whether it be online or hard copy, you were looking at that. We will see. All right. With that said, they're also at a relatively near point in the future. I will post some sort of extra credit stuff to bump up some scores a little bit. If you feel like it should be a little bit better. All right. So they're also, and again, as I'm talking about all of this stuff, there will be some curbing that happens with the test, but I can't tell you any of that stuff right now because I'm not ready to, all right? There was, as I evaluated stuff real quickly, I noticed one person had a 100. That does not mean that there is no curve, all right? So, any questions about it? Yes. Well, it's so that is, I mean, technically that's your grade. What it is likely there will be a little bit of a curve, probably not huge. And so that I will tell you, you're not going to see that change on Moodle, right? Maybe there is a way that I could get in there and make it work how I want it to work, but I don't know how to do that right now. I can say, oh, this test that should have been out of 100 points was out of 94. Anybody that got above a 94, now all of a sudden they got a 94, right? That's not fair to those people. If they get, you know, if there's going to be a curve and a little bit of a bump, if you got 100, well, you should you should still get a little bit of a bump as well, just because you got 100, right? Now you're looking around, you're like, who was that person, right? So you will not, when I will tell you what the high score is, and then that ultimately is what you will take, whatever you see. You know, if you've got an 86, you take an 86 out of 94. If I guessed that was your true score, I had to go buy a lottery ticket. All right, so is that any other questions? Okay, so yes, what you're seeing on Moodle should be really where you're at. And I can see all of that stuff. But like I said, I'm still not 100% ready to say all of those details. Okay. So this next chapter, I think is pretty awesome. There are some pretty cool images in here, and I know I don't think I have them embedded in the lecture. That would have been smart. But I do know with the ebook, if you are going through there are a couple of links that will take you to a few videos. Potentially, we will maybe visit some of that if you're today or tomorrow. We will see. I looked at it this morning as I was scrolling through, and I was like, oh, I wonder if this is good, but I did not open it up. All right, so there definitely are some embedded videos in this chapter, and I know others as well, that will give you some details and some animations that might help to walk through some of this stuff. All right. With that said, all right, and looking again, going back to the test, if you feel like you are struggling, if you feel like there are issues, I have office hours, even though they're not truly posted on there. It says by appointment. Feel free to meet with me if you would like to go over stuff. There are online tutors. If you look on the page, I think it should say tutoring. You can also go to the content area tutoring in Rendezvous, but I think a lot of it they have pushed over to Moodle. Not 100% with that. You've got access to free tutoring that way. And there are numerous other resources and online platforms that potentially can guide you through stuff as well, right? If you are not familiar, there is a website called Khan Academy. I believe it's khanacademy.org. It is full of all sorts of good stuff, all right? It doesn't matter, well, it does matter. There is some stuff that it doesn't cover, right? But it has all sorts of details. It has tutoring for pretty much any math subject. It's got lots of biology stuff, right? There's all sorts of other things as well, all right? I believe the story goes that individual, he was a professor someplace, his niece had some math issues, so he was trying to help her with some YouTube videos, and all of a sudden, there were like millions of people watching his videos, and it turned into this nonprofit where they basically go through all sorts of different books and walk you through math, biology, whatever it is, all right? And it's quick, 5-minute YouTube videos that walk through mitosis or meiosis. We're going to look at some of that stuff as we start going through it, this unit, all right? So that also could be beneficial to you guys if you feel like, seeing it a different way or hearing somebody else go through this stuff might be beneficial, all right? So there are numerous options, right? And definitely, tap into me. Your TAs hold office hours in room 208. They can help with lecture stuff or with lab stuff. They should be able to. And there are other people that man that room as well. If that door is open, it doesn't matter who that person is, your TA or not. Anybody that is at that table should be able to answer non-majors biology questions, all right? If they don't, there's a stinking issue, all right? So feel free to utilize them as well, all right? And just because you walk in there and they say, I teach A&P lab, that should not matter, all right? And if they try to tell you to go away, let me know and I will make sure that doesn't happen. Okay, are we good? Okay, I know there are concepts in here that are pretty detailed, pretty complex. And again, trying to kind of distill it down the littlest bit. I'm going to get rid of that. To make it a little more understanding, there's a handful of images of some cells that, you know, I'm a microbiologist. I like looking at stuff through microscopes. I don't look at things through electron microscopes, but when I get the opportunity to see them, I'm just like, holy cow, that's awesome. So there are a couple of things in here that we want to look at and stuff that we'll kind of talk about as we progress along. I'm going to point it out real quickly. So here we're looking at some cellular replication, right? And ultimately the formation of our little sea urchin in figure C, all right? So initially we start off and we're in the process of replicating over here, all right? We start off with a single cell. Truly, we start off with an egg and a sperm cell, both of which are haploid, and we will talk about this as we move through. The numbers of chromosomes get reduced in a diploid individual from 2 copies to 1 copy. And in the sex cells, the eggs and sperm respectively, they only get 1 of those copies. Upon the fertilization, upon the union of the egg and the sperm, now you get a cell, 1 cell that is diploid. It's got both copies of the chromosomes, all right? And then if we're talking multicellular organisms, we start going through mitosis, and we're going to go through details with what's up with that as we progress along. But it's ultimately cellular replication, all right? The cell is pulling in nutrients, it's metabolizing, like stuff we talked about before, right? And then as it has ample nutrients, it's ready to replicate and generate another cell, right? So in this case, we would have started off with a single cell that was the union of that egg and sperm, diploid, and it starts the process of replicating. So now we're in the process right here of forming 2 cells, right? These cells are replicating again by mitosis, and we'll go through this process. As we've got it on here, this image shows the cells that are in the process of constricting, pinching off to form 2 new cells or daughter cells, because that's usually how we call it. The one cell we call the mother, and as we pinch off into 2 new cells, 2 daughter cells, all right? There are a handful of proteins that kind of form the midline of the cell, and they start to constrict, to kind of pinch off. We call this cytokinesis, where the cell constricts and pinches into 2 new cells. And we can see it right here, kind of dimpled in, pinching off. That's kind of the last step of mitosis, all right? So in this case, you know, the sea urchin starts off as the single cell replicating, as we see in figure A. Over and over and over, this is happening until we get to the actual organism. So after a handful of rounds, there are 16 cells in this next frame. It's not as simple to see them all like we have right here because they're all stacked up, and it keeps compounding, all right? After many rounds, we end up with billions of cells, or potentially thinking our own bodies, trillions of cells that make the multicellular organism. It really depends on what you're talking about, who it is, all right? So the sea urchin, composed of all of these cells, all right? And there's all sorts of differences when we think about multicellular organisms. We talked about a little bit about prokaryotic cells and eukaryotic cells and some details of that stuff and different organelles. that are specialized in a eukaryotic cell. Well, when we think about multicellular organisms, we think about organs and organ systems and specialization with all of that as well, right? And we're not really thinking about that right at this point, all right? So we want to think about some similarities and differences between the prokaryotes and the eukaryotes, the bacteria and everybody else, right? We'll talk about some of this genetic material. We'll look at chromosomes. The DNA is similar, all right, but circular in the prokaryotes and linear in the eukaryotes, and we'll make some comparisons that way. We'll look at genes and traits, all right, as we start moving through and thinking about the genome initially, all right? So there are all sorts of potentials that help us to visualize this material. Different types of staining techniques that lend to kind of a tie-dyed appearance as we look at this cell. So really, there are a few things that we've got on here that we kind of want to think about, right? Initially, disregard all of these are chromosomes, disregard all of these guys right now, and just look at this, right? That is a cell, right? And you can see all of our colored chromosomes that have spilled out. This is with them still inside, right? And really what we're looking at is the nucleus of a cell, right? With all of the chromosomes all just in there, right? And there are, as we see this tie-dyed colored nucleus, there's all sorts of different cells. If we think about our own bodies, right? A skin cell and a liver cell and a kidney cell, they're all cells, but they're different and they have different functions, right? They all have the same DNA. They've all got the same chromosomes inside of them. They've got all of the instructions to make you, all right? But in those respective cells, within the nuclei of those respective cells, only some instructions are going to be used, and other instructions, well, those suckers aren't needed, right? You don't need that, right? Maybe you do some cooking at your house, right? Or you know somebody that does. They've got a cookbook, right? They use that cookbook to make all sorts of different recipes. But just because you have this cookbook doesn't mean that you make every single thing in the cookbook, right? There are some things that maybe you like more than others, right? Maybe this chocolate cake is your favorite, right? Whatever it is, all right? So some stuff is used more than others, all right? Dependent upon the type of cell, all right? So with that said, as we see this tie-dyed nucleus up here, if we were to put a handful of different types of cells and stain up the nuclei within there, you would see the coloring on these guys different, right? A skin cell may have more blue than green, and maybe a kidney cell would have a bunch of orange in there and hardly no blue or green. It really kind of depends, all right? So with that said, all of these chromosomes inside of the nucleus need to be dealt with. So we know which pages of that recipe book we want to be using, all right? So some stuff gets pushed out toward the periphery of the nucleus, kind of the colors we're seeing. That's the pages we don't really want to read, right? And the stuff that we want is more to the inside, where we're more accessible and we can open up those pages and we can read them, right? We can start making whatever it is we want to start making, all right? So I like this image because it shows us this nucleus. I kind of would like it better if we saw a couple of different ones so you could see what I'm trying to tell you. They all would look different depending upon the type of cell that you're looking at, all right? And it's not just a bunch of colors slapped up there to make it look interesting, all right? It's different types of genetic material, chromosomes, that stain differently based off of specific signatures, right? The assortment of the nucleotides lends to different dyes giving these specific colors, all right? Which I think is pretty cool, all right? And they've got all sorts of potentials with the fluorescence dyes to help assort and figure out these pairings as we look through here, all right? So first off, the nucleus, all right? What we are looking at with all of the other stuff is what we call a karyotype. And this is basically showing a picture, a snapshot of this nucleus and saying, these are the chromosomes, right? There are a couple of different potentials when we might think about this. Potentially genetic screening. Maybe you're curious about a bun in the oven and you want to know what sorts of details. Are there any sort of genetic issues? Too many or too few chromosomes can be super bad news, right? And doing some research like this shed some light onto that, all right? So a karyotype basically takes the chromosomes out of that nucleus, and then you have to do the work and go through and sort this stuff out, all right? Having different chromosomes with different stains dramatically simplifies this process of sorting, all right? You can stain the nucleic acids, the DNA, and they can all look kind of purplish. And then you just have a whole boatload of these things that are all purple or deep kind of blue. Then based off of size and shape, you can kind of pair them up, all right? This way is considerably easier because you can go through and kind of pick them out. There are some, you know, multiple colors that we could see maybe replicated, lots of greens, right? Whatever. So back to that picture of the sea urchin, I mentioned the egg and the sperm, both of which haploid, fusing together to create the diploid individual. Two copies of all of the chromosomes. And that's what we're looking at here as we've paired them up, right? They're all poured out there. They're not together when they, you know, are loose in the chromosome, but we pair them up for record keeping, to count, to see what's up. Are there differences? Are we missing, or do we have extra, or is one considerably shorter or larger than the other copy? That sets the stage for the possibility of all sorts of issues, all right, genetic problems, all right? So as we look at this, and we're looking at a human, we've got all of these chromosomes lined up. There are 22 chromosomes that we call your normal body chromosomes. We call them autosomes. These dictate lots of just normal details about you, all right? The last one over here, we see two of them that are the same. Although this one's got a little bend in it. Two that are the same. This is your sex chromosome or sex chromosomes. Two that are the same, X, female, right? If we had one that's larger and one that's small, almost like this 22, the Y chromosome is really kind of little. A big one and a little one, that would be male, XY, right? So really two X's. We just show a single X there, but really there's two of them in there, right? So from a karyotype, you can get these types of details and see if there's issues. You also can determine the sex, and you can see two X's, an X and a Y, or there are other potential issues. As we started this semester and we talked about some of these other details with extra or missing chromosomes, you can see through a karyotype these problems, right? If there are more or less, if you've got multiple X's or X and Y's, whatever it might be, a karyotype lends that information, right? And when you're doing genetic screening, this is the type of stuff they're looking at, right? You can't tell by looking at this stuff if there's been a mutation. Mutations are random changes in the genetic code with a nucleotide being replaced with a different nucleotide. You can't look at that and say, oh, there's a mutation that's occurred there, because the chromosomes all basically are the same size, right? But if something has been removed, broken off, and maybe moved to a different chromosome, we call it translocation. You can see that, right? These are genetic anomalies where chromosomes get manipulated, broken, and rearranged. Karyotypes can tell you that, all right? Now think about that as an example, all right? If number 18, a chunk of it gets broken off and put on 20, all right? I truly don't know any details that would happen to the person, but let's think about a cookbook, all right? Now we've got this recipe for the chocolate cake, right? And we're going through there and we're following this recipe. And then all of a sudden, the mutation over here, what should we make that's gross, right? What would you want in there that would not be good in your chocolate cake? How about some, what could we have? How about some roasted Brussels sprouts? Who the hell likes Brussels sprouts? I technically like Brussels sprouts because they're like little teeny cabbages, and I feel like a giant as I eat them, right? So all of a sudden, in the middle of my chocolate cake recipe, I throw in some roasted Brussels sprouts, all right? Deep down, that's probably going to be a delicious cake, all right? No, it's not, right? You got a huge issue. Or what if it's maybe a little simpler? Your chocolate cake probably calls for a boatload of sugar, and now there's a mistake up in there where instead of sugar, we put salt, right? That's not going to be good. That's really more of just a simple mutation, all right? We don't recognize that. But slapping the Brussels sprouts in the middle of your cake, that's a bigger problem, right? A chromosomal anomaly, all right? So karyotypes have the potential to lend some light on the subject, but they can't truly tell you everything, all right? Like the mix-up of the sugar and the salt. You may not detect that just by looking at the chromosomes, all right? Does that make sense? Hopefully so. So a little more detail about those chromosomes, all right? So as they're all paired up, all right? One from your mom, one from your dad, the two together are your homologous chromosome. So chromosome number 1 as an example, all right? The two pairs, and in a diploid individual, 2 copies, that's what you get, right? So on these chromosomes, and I'm gonna just back it up real quick here for just 1 second. These right here are all paired up as the homologous chromosomes. So the yellow 1s, or the red 2s, or the blue 3s, 2 of them together, all right? These are the homologs, all right? So there are differences and similarities that we can see on this, all right? So you've got, your specific chromosome, and it's got instructions for whatever it might be. Hair color, eye color, height. It's not just a single gene, that's lots of these things, but some things, all right? And one chromosome may say one thing, the other chromosome might say something different, or maybe they both say the same, all right? It's going to depend on what you have, and that ultimately makes you who you are, all right? Based off of the specific genes that are on there, all right? So you've got 2 copies. Each of these copies have genes on there, and dependent upon what those genes say, dictate truly who you are, all right? Same or different, whatever it might be, all right? So the locus is the specific position or location on that chromosome, all right? Where we might expect something like that to be, all right? And there are some potential issues where maybe some of these things get broken and reassorted, something like that, that runs to other issues, right? If you chop the instruction out and put it on a completely different page or chromosome, right? So each of the copies of your homologous pair originate from a different parent, right? And so your genetic assortment is the result of the 2 copies, one from each of your respective parents, right? So the genes or the copies don't necessarily have to be identical, right? And ultimately, that's a good thing, right? That lends to variation, right? As we look at everybody, it's easier for me to see everybody. Huge variation, right? You are all individuals, right? Genetically, if we were to look at the details in our DNA, we are all very, very similar, right? It's subtle differences in the DNA, subtle changes that ultimately gives us the variation that we see that makes people individuals in the first place, right? And that also becomes important when we think about sexually reproducing organisms. Variation is a driving factor. We need to have that variation in order for change to occur, for evolution to happen. There must be variation within the genome. And if there is no variation, you can't change, you can't adapt. You need to have variation, right? And it's, for the most part, really pretty easy to see when you think about people. When you look at other things, maybe it's not so easy to see, right? It really kind of depends on the situation with that. Okay, we're not going to get bogged down with that right now. So we want to think about how the cell goes through the process of doing all of the stuff a cell needs to do in the first place to ultimately replicate, right? We're not going to go through the details of pulling in nutrients and breaking stuff down. We technically already hit that stuff with metabolism, right? We didn't provide all of the specifics, but as we are pulling in these substances and breaking them down, getting energy, we also are producing these building blocks that we can use for growth and replicating these cells, all right? So all of that stuff we've already really kind of hit on, all right? We got ample nutrients, we're doing all of this stuff, we're making the building blocks. Now, how do we deal with it, all right? So the cell cycle, we'll go through and talk about the specifics with it, the steps and stages. When we think of this, we're talking about eukaryotic organisms, all right? We will talk about the prokaryotes, the littlest bit. They do replicate, but it's simpler because they don't have all of the extra organelles. They don't have boatloads of chromosomes. It's a lot easier for these guys to replicate, all right? So we'll talk about details about the chromosomes, what's happening to them in mitosis. You should, in the very near future, I believe, be looking at some of these cells in the lab. Is that true? Do we know? I see somebody shaking their head. No, they're like, nope, I don't know. It should be coming up pretty soon, I believe. All right. So you get a chance to kind of see some of this. So we'll look at that and we'll look at some of the other steps and ways that we can govern whether this stuff happens or not. You would hope that your cells aren't just constantly dividing, all right? You don't truly want that to happen. That's more of like a cancerous situation when cells are just going haywire, all right? So there's some control mechanisms, some kind of checkpoints, if you will, that ensures that things are happening properly in the cell, all right? And I guess, since I already mentioned it, really deep down, when cancer happens, it's a lot of this stuff that's getting messed up. These checkpoints, these control mechanisms. Now, all of a sudden, we're like, I don't care, right? I don't care if you've done it or not. Just go ahead and replicate, right? It's all fine and good. That lends to bigger problems, right? And there's other stuff as well. We'll see a little of that shortly. So the cell cycle, it's a handful of steps, ordered steps, that ensure that everything is proper in the cell in the first place, so that when it does replicate, the two new cells are good to go and have all of the stuff inside of them that they need, all right? So we see cell growth and then ultimately division. Again, pulling in nutrients, metabolizing, breaking stuff down, and getting these building blocks. That ultimately sets the stage for recognizing we're good to go, right? We're ready to replicate. I got all the ample nutrients that I need. I can replicate my DNA, and in turn, we can make two new cells, right? Ultimately, with all of the stuff inside of these cells, the mitochondria and the endomembrane system, all of that stuff, it gets replicated as well. And that's happening when we are just pulling in nutrients, when we're down here in what we call interphase, right? That's when the cell is building all of this extra stuff. And then it says, we've done it. We've got all of it we need. I've made room for more. Duplicated all of these things. Let's split in half, all right? So interphase is when your cells are just hanging out being normal cells, whatever kind of cell they are, all right? Most all of our cells are in interphase most all of the time, all right? And then we see a little bit of replication. Some of our cells get generated and they stay as that, stuff like nerve cells, as an example. And I skipped over, but the G0, the G0 is where they just hang out like that, and they're not going to go through the mitotic phase, right? They're not going to replicate. So all of this stuff happens. The cell grows, the DNA gets replicated. So we call that the synthesis phase. And once we have done that, then we move into the separation, all right? Mitosis, once we have initiated it, it should go to completion, right? We shouldn't need to wait, all right? There are some potential issues with that, all right? So in mitosis or the mitotic phase, the DNA that's already been replicated and along with those cytoplasmic contents, all of the guts of the cell, they get separated into two roughly equal halves, and that cell divides, right? And really the process of mitosis, we talk about it to think about how we move these chromosomes to ensure that all of these copied pairs get partitioned so that each new cell is genetically identical, right? Mitosis gives you cells that are the same, right? That's important as well, right? They're genetically identical. They're clones of one another, right? Unless there's been some mutations, then there's issues, right? So with that, we can see this detail trying to show us the process of the cycle, the cell cycle, circular, all right? All of this stuff in gray, all of that is interphase. And the littlest bit of that pie, this little bit right here is mitosis, right? And we're trying to show it this way to let you kind of understand the bulk of the time, it's interphase, right? It's only a small little piece of the pie where the cell is actually just separating apart, right? At this point in mitosis, it's not doing normal cellular stuff. It's not pulling in nutrients. It's not doing that. It's separating, all right? The rest of the time is when it's just hanging out being a normal cell, right? There are some other things happening, but still a normal cell. So with interphase, we separate it into 3 bits. The first one, G1, this is a first gap. This is when the bulk of the time is spent metabolizing, pulling in nutrients, and making building blocks. Once we've got an ample amount of the building blocks, the nucleotides needed to replicate the DNA, that's when we move into the S phase. S for synthesis. This is where the DNA gets replicated. right? All in this growth phase. The first G1 phase, this is where we're replicating other stuff, organelle material inside of the cell, pulling in ample nutrients, replicating that stuff. Now we got a lot of extra nutrients, we replicate the DNA, all right? In the synthesis phase, that happens, we will go through DNA replication later, all right? So it gets replicated. Now we've got a chromosome that's replicated, it's paired up like this, all right? And we've got the other chromosome replicated, paired up like this. They look like two little X's linked together, all right, by a protein that makes it look like a little X, right? That's a replicated chromosome, and we call those guys sister chromatids, right? When they have replicated, right? And the two replicated sister chromatids that are the same chromosome number, we call that a homologous pair, right? So all of that stuff happens. We synthesize the DNA, and then we move into the last bit of interphase, G2. This is another growth period, and really it's just kind of preparatory for the cell division. So we've got a cell that's basically the same size as all of the other cells, pulling in all of the nutrients, replicating stuff, replicating the DNA, and then the second gap, it gets bigger, increasing the cytoplasm so that now we've got about 2 cells worth in a single one that pinches off. And now we've got 2 that are about the same size that we started with, right? So the second gap is just increasing volume, really, so that we get cells that are about where we started right here, right? So again, lots of stuff happening in interphase, and the bulk of your cells are somewhere in this process at any given point in time, right? And then they hit mitosis, and this is typically a pretty rapid process for that cell to separate the chromosomes and then go back to 2 new daughter cells, right? A very short window of time for this to occur, all right? And it also kind of depends on the type of cell, all right? So here we've got a couple of images that try to put this into a little bit of perspective. What we are looking at is that little chunk of the pie, right? The mitotic phase. We're not looking at interphase, just mitotic, all right? So with this, there are a handful of different steps. Things are happening to the nucleus. Things are happening to the chromosomes. And we can witness that stuff for the most part as we progress through this cycle, right? And this, what we're looking at is animal cells. We'll show you a plant cell. It's similar. There are a few differences with plant cells, how they go about replicating, right? So, a couple of different steps, and this figure, it's kind of hard to see all of the details, but really the cartoon or the fluorescence images that we're looking at below are really kind of what we want to think about in the first place. So as we start, we are coming out of the second gap, the tail end of interphase, and that brings us right to this point. As we hit this bit, there is a checkpoint that ensures we're moving into this. The first bit is called prophase. And during prophase, if you were to look at these cells under the microscope, you're going to still see the nucleus. If you look at them in interphase, at any point in interphase, you see a big purple nucleus, right? Prophase, you still see that. The nuclear membrane is still intact. You can't see the chromosomes. It's all still harboring all of that stuff. And that's what we're seeing right here, all right? The nucleus. You can still see it up above, all right? That membrane starts to degrade, all right? The nuclear membrane starts to kind of fall apart, opening it up so that we can partition the chromosomes. Toward the tail end of prophase, It's virtually all degraded. And that moves us into this kind of in-between step. We call it prometaphase. And it's at this point that technically you could start to see the hints of some chromosomes, right? Not in prophase, but prometaphase possibly, right? In prophase, the chromosomes start to compact and condense so that they're nice and neat and easy to separate, all right? And in prometaphase, they're kind of continuing to condense, but the membrane's now degraded. There's a bunch of these little guys up in here that are microtubules linking up to these chromosomes. It's like fishing line that's going to try to pull them to opposite sides, all right, as we reel them in, all right? So down below, we can see the hints of some of this. And it's really kind of, at this point, it's still lots of bluish, purplish stuff, but these are a bunch of chromosomes that are all of these green guys are these microtubules that are linking up to the chromosomes. So at this point, we're kind of in a tug of war, getting stuff positioned and trying to link up so that we can start this process. As the spindle and the tubules attach, now we move into the next bit. This middle bit, we call it metaphase. And the chromosomes, this is when, to me, this is when they really are visible, all right? Like I said, you potentially can start seeing some stuff, but usually it's kind of just all over. In metaphase, they are all nicely positioned. The tug of war has pulled all of these homologous pairs to the midline of the cell, all right? They're all linked up. We've replicated all of this stuff, and now we just want to separate what we've replicated to opposite sides, all right? So metaphase, they're all lined up along the equatorial plane. Here we've got them going vertical. They could be horizontal or any, dependent upon the plane of division of those cells anyway, but they're in the center, all right? So all of the green guys start to contract. That brings us to anaphase, where we pull these pairs apart. And now the sister chromatids, the replicated chromosome number 1 from your dad, gets separated like that. And the replicated chromosome number 1 from your mom separated like that, all right? You have 23 pairs of chromosomes. And these 23 pairs, they all replicate and they all line up in a nice row and separate like that, all right? That's mitosis, so that each cell has the exact same stuff as the other, all right? So they separate them. You can see these guys getting pulled to opposite poles as they get toward the end. That brings us to really the last bit of mitosis. We call it telophase. And in telophase, at this point, it's almost like prophase. You'd be pretty hard pressed to see individual chromosomes, right? The nuclear membrane starts to resurround the individual chromosomes that have been pulled to the poles and makes it, it's like a veil or a shroud that we are putting around those chromosomes. It makes it super hard to visualize them at this point, even though here you can still kind of see it. It's the fluorescent dye that lends to that, right? So it's surrounding it. The chromosomes start to kind of decondense, uncoil a little bit. We're going to open them back up for business, right? As we're going through this process in prophase, we're packaging them up super tight, right? It's like the book still has shrink wrap around it to keep it nice and neat, right? And here we're unpackaging it so that we can actually read those recipes, right? And then, I really don't know why, but cytokinesis, they don't put it as part of mitosis. It's got its own little different color right up here, right? It happens at the tail end, and they say it's separate, all right? I don't know why. I feel like, can't it just be the end? It seems like it should be. So cytokinesis is the last bit, all right? Whether we really call it part of mitosis or not, I guess is beside the point, all right? So cytokinesis technically starts happening towards the very end of telophase, where we start seeing a little bit of constriction back to that sea urchin cell that we could see pinching off. That's what's happening right here. And a handful of proteins start to constrict, bringing the cytoplasm to the midline and ultimately pinching it off into two new cells. Genetically identical, all right? I kind of sound like a broken record, all right? But it is important to realize mitosis is the same, all right? If you start with a cell that's got 46 total chromosomes, when you are done, you get two new cells that both have 46 chromosomes. They are the same, all right? Skin cell and a skin cell, or whatever it is, all right? That's important, all right? And that little sea urchin, as we start with the sea urchin, once that haploid egg and haploid sperm fuse, we go through mitosis very rapidly until we hit the adult stage. And then still mitosis happening, but we need to think about the generation of sex cells, eggs and sperm cells, and we need to think about going from diploid to haploid. That's a completely different process, but it is very, very similar to what we've just talked about, but twice, right? As we go through this twice, you can go from 2 diploid cells to 4 haploid cells, right? 4 cells that are unique, right? And that we will talk about later, all right? But it's important to recognize there are differences with that, all right? And mitosis is super important because the bulk of all of our cells, that's what they're doing in the first place. We only talk about differences with your gonads, right? So cytokinesis, this is a little bit different within plant or animal cell, animal or plant, I should say, all right? We still have these divisions, but it's a little different, all right? So with this, and we saw the sea urchin, it was a pretty good image of that. As the protein starts to constrict, it creates this little cleavage furrow, this pinching in, and it continues to, the proteins continue to constrict, giving us 2 separate cells, right, when it's all said and done. In a plant, it's a little bit different, all right? So there are still, you know, the plant's got cell wall, it's got all of this other stuff inside of it that some of our animal cells, it's different. Chloroplasts and vacuoles and things like that. We don't care about that, all right? The cell wall is important, all right? So initially we start off, and this is already in the process of dumping it in the middle, all right? It starts off as a large cell. We've separated the chromosomes to opposite sides. And then there are little bits of vesicles that start dumping cellulose, basically. New cell wall is getting laid down, right? So Golgi vesicles with their packaged up cellulose start moving toward the midline of the cell, and this starts forming the cell plate. It's not cytokinesis, it's not a bunch of proteins that constrict it, it's vesicles inside that basically lay down a wall between these 2 cells, right? Here it looks like just a single wall, but over the course of time, it becomes 2 separate, distinct cell walls. They each have their own wall, right? It's a double fence, right? And if you recall, there are little pores in between the plant cells called plasmodesmata that allow for communication in between, all right? It's not a complete, impenetrable wall, right? So similar, but a little bit different, right? And some of the proteins needed to separate the chromosomes also a little different, but for what we think about, basically the same, right? But cell wall versus plasma membrane requires an extra kind of set of vesicles to deposit that material. So I mentioned a nerve cell a minute ago. Some cells don't continue to divide, and they go into this G0 phase, or what we call arrested development, right? where they stop replicating, all right? So some cells may be in arrested development and move in to the cell cycle, and others, like a neuron, potentially not. They are in this G0 phase indefinitely, all right? With that said, there are researchers trying to figure out some of this stuff, like neuroscientists trying to figure out things like spinal cord injuries and other neurological problems. Can you turn these cells back on? Can you make them replicate? Could you potentially repair something like that? And I'd like to think we are so close to having these capabilities, right? There is tremendous promise with this, right? And possibly lending to, people that have been paralyzed being able to walk again if we can target the specifics and repair some of this damage. I think that is fascinating. So it really kind of depends on the circumstances, right? Where some might be in this process, right? And that's really it. Oh, I did put a link in here. I knew it. I knew I must have. All right. Or I had hoped. Okay. Now I'll click it and I will have wished I never did. So as we look at this, it's the same cycle that we already kind of talked through. But what we're putting in here are some of the extra details, all right? So there are a handful of boxes, a handful of checkpoints that basically it's cellular surveillance. We want to know that stuff is done properly. So when the cell does replicate, we don't have issues, right? So these checkpoints really help to ensure that we don't end up making mistakes, right? And with cancerous situations, some of these checkpoints get messed up, and that lends to, abnormal growth, right? So this very first point, we call it a restriction point, right? This is our first stop sign, and I like to kind of think of it like a crossing guard, right? You got a bunch of little kiddies waiting to go to school. The crossing guard helps to get them across the street. And when the sign is up, you have to stop, right? So this first checkpoint ensures that our cell has done everything it needed to do. Did we replicate all of these organelles? Do we have a boatload of mitochondria now? Are we ready to do this? Do I have ample Golgi and all of these other things, right? If all of that stuff seems good, there's also ample nutrients. I got all of the nucleotides. I got all of my ingredients. I just went to Winco or wherever you are shopping, right? And I got all of my ingredients to build my cake, right? To bake my cake. You're not building a cake. Maybe you won't. So I've got all of this stuff. My first checkpoint is basically doing some surveillance and saying, I do have all of the things I need. It's called a restriction point because once we cross this point, we're going to continue, right? It's committed after that. We're going to synthesize DNA and we are going to process this. But at this point, maybe we don't quite have all of the ingredients. I thought I had sugar, but I went to the cupboard and all I have is salt. I still need to go back to Winco. So I'm going to put the brakes on baking this cake till I go get the rest of my ingredients. And then I'm going to come back and do it, right? This first checkpoint is one where we can stop and we can finish building our stuff or gathering our nutrients. Once we have moved past this first stop sign, if something else has happened, you're kind of out of luck, all right? This is an issue. And so you hope that this crossing guard knows what's happening, right? I didn't see the stuff. My wife just showed me a quick picture of this. I don't know where it was, but apparently this place, this little town, their crossing guard was a rooster or a chicken. And it had probably a chicken, because roosters are mean, and I think they would chase the kids. This little chicken had a little vest on and she was walking the kids through why did the chicken cross the road? To get the kids to the other side, right, is what I figured. Checkpoint, all right. Does that chicken know what's going on? Probably not, right? So we move through if everything's good and the stop signs drop. We replicate the DNA. We go through all of this process, making sure that we've got all of the chromosomes copied. As we move through the synthesis phase, you see there's no stopping here. We do this, we replicate, and we move up into the next step, the second gap, right? And this is another checkpoint. But in this case, if something has happened, if we didn't properly replicate the DNA, we don't just say, oh, we're going to stop and wait till the stuff gets fixed. It's already synthesized. So if a mistake has happened, it's a dead end, right? And that cell stays with the stop sign up, right? It doesn't progress along, right? That in turn is going to create some other issues, right? We have tried to do stuff, but now other cells see this flag and they say, wait a minute, what's up with this cell? There must be a problem. And we target it for destruction, right? We're not going to let you persist. We will destroy you, right? So that's a mechanism to hopefully ensure that we don't make a bunch of cells that are all wacky, right? If everything seems fine, the DNA has all been replicated, so we got all of these sister chromatids, they're all hooked up, all of that seems good. Then the next stop signs drop and we move into mitosis. We start going through this, and the last checkpoint, this M checkpoint, is the metaphase checkpoint, and it ensures that we have everybody lined up at the midline. Again, if we don't, there can be the littlest bit of lag time to let somebody keep reeling it in to get them all in there. But if it ends up that something is messed up and we don't all make it to the midlines, again, the flag is up and the cell gets destroyed, right? If everything is happening properly, we finish the replication, and now we have two new daughter cells that go through this again, all right? You want me to click on the link? You want to see it? Okay, I see two yeses. I looked to you because you shook your head before. You said no, right? I can't remember what it was. No, about the lab, right? Not you. You're like, what? Don't put me on the spot. Okay, let me see if this works without. I bet you I need to stop my share and share this. Sisters, why didn't we just watch that and not hear me spouting off? Okay, questions or concerns? All right, guys, have a great day.